Oh, you're good. I was just going to say it is officially noon. So, welcome everyone to our weekly Iowa Learning Farms webinar. I'm Elena Whitaker. I'm the Water and Conservation Educator with Iowa Learning Farms. And today we're joined by Dr. Uh, Roland, Roli, Wilhelm. He is at Purdue University and he's going to be talking about soil microbiome and biological testing. Where are we now and where are we going? As usual, if you have any questions, feel free to type them in the chat throughout the presentation and we'll get to them at the end and we'll do a quick Q&A. So with that, the floor is all yours. Go ahead.
Thank you, Lana, for the introduction. So saddle up, everybody. It's rodeo season. I'll be presenting a number of slides, and I erred on the side of offering, at least you get to see the slides, and if you want, you can come back to it. But we will be moving relatively quickly through this. I like to start with this quote. It says, "Do I contradict myself? Very well, then. I contradict myself. I am large. I contain multitudes." And I say to any farmer, let your soul stand cool and composed before a million universes, because that's what Walt Whitman would have said about the soil microbiome. And that's partly because we are large and we do contain multitudes. The theme of this talk, in some ways, is that we live in a microbial world. We large organisms with many, many different compartments and cells are coated with microbes. They live inside and all over our bodies. And you can see here on the right, there's a picture of a root, a lateral root emerging from that main root, and it's stained in red. And all of that cloud of green are different individual bacterial cells. That sort of armpit of a root is actually where some of the sugars leak out. And so this is usually one of the hot spots where microbes colonize and grow on your plants. And these untamed multitudes, the multitudes in your field, there are so many of them, partly because there are so many different little habitats for life to occupy in your soil. So you have, at this scale, you have the many organisms that you can see with your naked eye. You can often see fungi growing. You'll certainly see mites, some large nematodes you can also see. But then if you zoom in further and further, in this case, looking at one of the surfaces of a soil particle in your in a soil mineral, you can see a sort of colony of bacteria living on that surface, and you can see them using resources and eking out their life there. And if you zoom in even further, you'll find viruses and all sorts of other smaller bacteria interacting in your soil. So there's a diversity of different habitats all the way down to these tiny scales where organisms are living on your minerals. And so, one of the takeaways that you'll find anytime you talk to a soil microbiologist is that your field is very, very likely to have a unique microbiome present. I visited 14 farms this past year in Indiana, and each one of them was different. And so just a quick overview of what my talk is going to be about. I'm going to give you an explanation of what makes these soils such unique biological habitats especially for microorganisms. Then I'm going to tackle some common questions like how do soil microbes contribute to soil health? And then how do I manage these multitudes? And then lastly, I'm going to touch on this question that I often get in terms of, soil microbiome testing. It's this new hot sort of offering that companies are selling to farmers. And the question is it worth it? So let's start with what makes soil biology so unique. I'm going to throw a couple of different big picture figures at you, different graphs that show where soil stands in relation to other common environments like humans, the ocean, or freshwater lakes. And so on the axis here, it's the number of millions of unique species. So you can see here in soil, we have the highest number of unique organisms, around a billion, 10 billion, no, a billion or so. And so soils are home to the most diverse array of organisms. And then what makes soil so unique? We also know so little about these organisms. So there's a billion different species, and of that number, soil, similar to lakes and marine systems, we know actually only about uh, what 75% of those organisms look like and what they do. So there's still this huge amount of biology to discover in your soil. Also, soils tend to be characterized by organisms that grow more slowly than other habitats. Perhaps unsurprisingly, organisms that live in or on our bodies grow quite fast because we're warm and they like that, and that helps them grow faster. But in soil, it's much more similar to lake where organisms tend to grow a lot slower. And then this is probably the most um, different aspect of the soil, and that is that the average genome size of an organism in soil tends to be larger than all of these other environments. And so when I talk about a genome, I'm talking about all the different genetic material that's encased in that cell. I often describe it as the instructions for life. So essentially, in soil, an organism needs more instructions to survive than in other environments. And to just drive that message home, I like to introduce this one bacteria called Mixococcus. It's a common soil decomposer. It's in everybody's fields. It's often found breaking down the residues left over after you've harvested your crop. Now, what's so special about it? Well, its complex behavior includes the ability to glide along surfaces. So hopefully you're seeing that video move here and you're seeing what looks like little snakes from your Nokia cell phone back in the early 90s. They're fluorescing, and you can see that they're sort of gliding along the surface of that slide there. And what's so special about that is that when people studied how that happens, it looks like they actually have, similar to a tank tread, the ability to sort of slide across the surface like that. And so that's an adaptation to all of the different surface areas in a soil. These same bacteria signal to each other when they discover prey. They actually feed on other bacteria, they're predators, and when they find prey, they'll release a signaling molecule that tells everyone, all the other Mixococcus in the area, to crowd together and come on in. Similarly, they've adapted this ability to make what are called fruiting bodies. So on the bottom right of your slide here, you see these sort of weird, spiky sort of structures there. And what each one of those is hundreds of thousands of these different individual bacteria forming into this superstructure. And you might ask, why are they doing that? And that's because getting around in soil, if you're the size of a bacteria, is really hard. And so what they've kind of figured out is that if they make this sticky, weird structure, it can actually stick to a worm or some other kind of burrowing insect or something else that's moving through that soil, and it latches on, and they're essentially hitchhiking. So each one of those fruiting bodies you can think of as like a sticking our thumb up and asking for a lift. And so in order to do this, this one bacteria has to have a pretty large genome to allow it to function like that. And so the other question is, so why does soil lend itself to this array of different life forms? Well, the first part is that there's lots of surface areas. There's lots of real estate for organisms to occupy. And there's also many neighborhoods. So this video I'm going to show next is a macro aggregate, and they've taken an X-ray of it in high resolution. And so you're going to see a cross section as we move through the aggregate. And I want you to pay attention to the different sizes of the particles in this macro aggregate. You see larger ones that might be more like an open plane, and you see a lot of small uh, enclosed clay particles that make it probably more like a maze, like a corn maze. And so each one of these habitats, each one of these locations within that soil is a habitat for microbes. And here's an example of that. We're seeing what's lighting up there are the individual cells of bacteria as we do that same kind of cross section through a simulated soil particle. Call it a transparent soil. And so these hot spots are actually bacterial cells finding different pores and living in those soils. So you have many neighborhoods and lots of real estate in your soil. And also, what makes so, oop, I don't want that to go again. Within each of those aggregates, you have this distribution of resources that's different. So one of the most obvious examples of that is oxygen. So from the outside, that aggregate is getting access to all the gases moving through the soil. But as you move closer and closer to the inside of that, all that oxygen is being consumed by different organisms to breathe and to survive. And so you have this gradient across the outside to the inside of that particle, and that also gives all sorts of different types of habitat and different advantages to different organisms. Okay, so in summary, soil biology will be contradictory at time because it contains a diverse habitat with very, very high number of different organisms. and then let your soul stand cool and collected before a million universes. That is advice I have to follow myself every day because we really have a fairly naive understanding of these multitudes. Soil biology, it's not new, but it's certainly still a young science when we start looking at how many of these different organisms we actually understand. All right, so moving on to part two of my talk, I'll address some common questions about soil biology and soil health, specifically how soil microbiome or soil microbes contribute to soil health and how you might manage these multitudes. So one of the most practical ways of thinking about all the different organisms living in your soil is grouping them into these broad categories that we call functional groups. One is called the biochemical transformers. And so these are the organisms that are essentially doing magic in your soil. They're taking one sort of compound and they're doing some transmutation and converting it to something else. They're incredible catalysts that can do a whole bunch of different chemistry. There's the second group are called biological regulators. And these are sort of like the trophic web, right? When you think of the or little guys being eaten by bigger guys, by being eaten by bigger guys, these are the organisms that are either predatory or parasitic, and they're part of this sort of web of interactions between smaller and larger organisms. And that what's pictured in that circle here is a nematode that will feed on bacteria in your soil. The last category, but not the least, are the ecosystem engineers. These are the organisms that can shape and reshape the structure of the soil. So classically, the earthworm is burrowing holes and redistributing organic matter from the surface below ground. But there's a whole range of different organisms that are creeping and crawling through your soil and creating pockets of space and also releasing sticky things that bind the soil together. So these organisms are engineering that environment. I don't have time to talk about all of those different groups, so I'm going to focus on the biochemical transformers today. And that's because they really do such an incredible job of transforming your soils. They're the organisms that are cycling all of those key nutrients that you provide or want your plant to have access to, like nitrogen, phosphorus, sulfur, carbon, and the list goes on, iron, et cetera, et cetera. So just as a maybe soil microbiology 101, it's the life in your soil that actually regulates every step of that nitrogen cycle. So when you add fertilizer, microbes themselves, whatever's not being taken up by the plant is being used by different microbes for different purposes. And those bacteria influence both the gain of nitrogen to your field. So you can see here, this nitrogen that we're breathing right now actually can be converted into a form of nitrogen called ammonia that's accessible to your plants and other soil organisms. And then that can actually be converted by other bacteria into nitrate, which again, very more accessible to the plants, but also more prone to leaching. But those are all gains from the atmosphere. And so that's biological nitrogen fixation that's sort of adding to your bottom line there. But of course, microbes giveth and taketh away. There are also a whole group of microbes that will convert that water soluble nitrogen back into gas that's no longer accessible to organisms and your plants in the soil. Similarly, microbes are releasing phosphorus from your minerals. They're mining mineral in your soil or in your fields, and that's because plants are not particularly capable of accessing it themselves. They form partnerships with fungi and bacteria that essentially dissolve and erode the minerals to release phosphorus that the plant can then use. So that's happening here. You can see an example of a petri dish, and that white haze that you're seeing is actually calcium phosphate, a common form of calcium in your soil. And the organisms, these are these little colonies here. And anytime you see a clearing around that colony, you know that it's actually solubilizing that calcium phosphate and not making it available to the plant. And so you can see that not every microbe in your soil is doing that. And I actually have a project on this in Indiana as well. And I was surprised at relatively how few of them are doing that. I mean, it seems like there is some kind of improvement we can do by adding these microbes to soils. Another thing that these biochemical transformers are doing is producing goop. We call this EPS or extrapolymeric substances, but essentially it's just sort of like a snotty, goopy substance. And that's because life of all types has figured out that water is important to retain. So what this goop does is sort of soak up that water before it dries up, and it gives the organism a little extra bit of protection from desiccation and drought. These organisms are typically photosynthetic and they'll live on the surface of your soil. I mean, all sorts of microbes will produce this goop, but the most prolific goop producers tend to have access to the sun and they're photosynthetic, just like your plants. And they'll also add nitrogen. So every time you step on your field and you see this green crust kind of forming, that's cyanobacteria and algae, and they're doing some important work for you. And of course, this goop helps bind soil together, just like it helps the organism protect itself. Okay, so that's sort of an idea of what these soil microbes and how they're contributing to the health of your soil. All that's happening 24/7, day in, day out. And so these organisms are there and they're basically your pets. So the question is, how do I manage these guys well? The good news is that following basic soil health principles, they're kind of designed to help the biology in your soil to begin with, right? So you have this USDA four principles of soil health. That is to maximize continuous living roots, minimize disturbance, maximize soil cover, and maximize biodiversity. And I like to break these up into two main parts. So one is feeding your soil organisms. The more continuous living roots you have in that soil, the more sugars are being shuttled below ground from your plants. And then maximizing biodiversity. That's sort of, you can see pictured there that there's some cattle roaming in the pasture. They're fertilizing those soils. They're killing or they're feeding on the plants, and the plants then regrow their roots, and they're sort of adding more nutrients below ground. They're recycling the nutrients. And then similarly, you want to protect your soil organisms. So minimizing disturbance and maximizing soil cover, that prevents those organisms below ground from getting chopped up as you till or disturb the soil in different ways. And then maximizing soil cover, that helps retain the moisture and keep the temperatures from getting too high, because soil organisms are just as susceptible as we are to temperature fluctuations. Okay, so beyond that, those four management practices, there are more direct ways that you can kind of manage the microbes in your fields. And one of them is called biostimulation. So that involves kind of creating the conditions in your soil that favor the growth or activity of native populations, the ones that you want to stimulate. And I'll give you some examples of that. The other part is the other a way to manage is called bioaugmentation. So that involves adding desired beneficial microbes to your soil or your field. And so one is essentially, feed what's already there, and the other one is add new organisms to your field to sort of make up for any lack in that case. So let's go through each of those examples. The most simple form of biostimulation is by keeping those roots in the ground. So having cover crops, and maintaining plants that have healthy root systems. The healthier the plant, the more carbon it's shuttling below ground. And that supports an active community. I have a really kind of a treat here. I'm going to show you a video. It's taken of it's actually an X-ray of a corn root that you can see on the left there. And what the researchers have done here is they've used a form of carbon that's called as a stable isotope called carbon 11. And with that, they're actually able to identify where that carbon goes and trace it from where they introduce it, which is above ground. And then after the plants have photosynthesized it and they've shuttled it below ground, you can track where that goes. So it's quite a remarkable video. And just keep an eye on how fast it happens. So at the beginning, you'll see within 20 minutes, you still see the cloud of carbon 11 at the surface. But then it's slowly heating up and being moved through that root system as that plant is fixing that carbon and moving it below ground. And you see that there's little blurbs at the bottom of the roots. So that's a great example of how much carbon is spilling out of that root into the soil around it. You can see these hot spots about after 2 hours now. That's how quickly carbon is being converted into sugar and shuttled to the roots of your corn plant. So I find that a fascinating video, and it's a direct view of what's happening below the ground that we often don't get to see. And that's, of course, biostimulation. That's providing all the organisms of all different sizes access to energy. The most classic example of bioaugmentation is with your soybean. So Bradyrhizobium are not necessarily native to North America, or at least the parts of the North America where we're growing them. And so we've had to add them to our soybean to help turbocharge. And I love this this data on the right. So there's a clear example here when you look at the potted plants, when you grow them with or without that bradyrhizobium, which of course is a root nodulating nitrogen fixer. So it lives inside the roots, and the plants trade carbon for nitrogen in that environment. And so if you look to the right here, there's all these green dots. And this is a really interesting, each one of these dots is a separate scientific study. So this is what we call a meta-analysis of a huge number of different studies. And on the bottom, you see this is effect on grain yields based on nitrogen fertilization. You can see that anytime you add nitrogen fertilizer, it pretty much boosts the yield that you're going to get. But not all the time. You see some dots below that zero line. And if you compare that to in this case, not changing the nitrogen fertilizer, not providing any fertilizer, just providing that bacteria, the bradyrhizobium, you also see a pretty stark boost in that grain yield. So this is just an example of when you add a microbe that's missing from that environment, you can see direct benefits to your cropping system. All right. So in summary from this section, how do soil microbes contribute to soil health? Well, they're regulating huge amounts of fluxes of energy and nutrients through your soil. The question is, how do I manage multitudes? Well, you can feed and protect them. You can also stimulate and augment them. Okay, the final part of my talk um, is on the topic of soil microbiome testing. And the question I'm posing is it worth it? I like to show this what I call the soil health tangle, because everything we talk about with soil health is often interrelated, and you change one thing, it can also impact the other. So in terms of the soil biology dimension to soil health, you kind of have three main things that get all tangled up, and that is the biological activity, who's alive and doing the work. You also have the availability of resources. What's providing the energy and nutrients for that biology to be active. And then the last one is sort of the simple, actually, oh, I haven't been showing. So for biological activity, when you do soil health testing, you often look at the respiration, how much carbon is being respired from your soil, and that gives you a proxy for how active your soil is. And you can also track the same kind of measurements for nitrogen. For the resource abundance, we do tests like the water extractable carbon and nitrogen. So this is the simulating what is easily and readily available for the microbes or the all living things in your soil, say after it rains. And then we also have this idea of how much protein is present and how much carbon is in that active pool that sustains life. And then the last one is kind of newer. This is a technique that depends heavily on DNA sequencing, and that is identifying who's all there in your soil. And we call that the composition of the community. And so this DNA sequencing piece is really what we're talking about when we talk about the soil microbiome. Yes, there are other ways of characterizing who's present in your soil, what the community looks like. PLFAs, which looks at the lipids of the organisms, has been around for many decades and is still used today. But the real push to this soil microbiome testing has to do with DNA sequencing. So I'm going to take a second to kind of walk us through what is going on in these commercial labs that are offering this test. So to begin, we can kind of imagine in our soils this diverse array of microbes. I've shown you some of them. Each of them have these inside of their cell, a little circle that symbolizes their genetic material or their genome. So what happens next is essentially we can characterize each of these cells just based on its genome. And so this is sort of a conceptual understanding of what that community looks like. It's all of the genetic material encoded in these different bacteria or these different organisms. What happens in the lab is they create what they call a sequencing library. So they break apart all of that DNA, they put it into a sequencing machine that does some pretty amazing chemistry, and they're able to translate from analog, the actual polymer of DNA, into the digital realm where we have a sort of a sequence code. We then take that sequence code and we can back calculate. So you can see here these different colors. This is sort of the abundance of each of these different organisms in that original community. And you can see that the blue one is much more abundant than the greener ones. And we can learn that from our sequencing library. We can also ask questions about how many different microbes were there in my soil. And then lastly, the thing that a lot of these soil microbiome tests aim to do is predict the function using a database. So they have a repository of all the information we know thus far about the 75% of microbes living in your soil. And we try to infer based on who's there, what they're doing. And I have a special little section on that because that's where this whole testing part gets a little bit complicated. And so let's talk about this idea of predicting function from the soil microbiome. So I just explained to you that this idea of the soil microbiome really comes down to just the total number of who's present in your soil. It doesn't capture anything to do about their activity and their environmental condition, right? And those two things are very important for what these microbes or these organisms are doing in the soil. So when we say soil microbiome, we really kind of mean one or both of these things, the who's all there and or what genomes are present in that soil. And just to reemphasize that, to go from a genome to actual biological activity, you first have to express those genes, which is sort of a messenger molecule. And then that messenger molecule has to be read to create a protein. And that protein is the unit that actually performs the chemistry. So without those two downstream steps, you can kind of see that there's a disconnect between who's there and who's got what things turned on, right? So we can't actually predict easily what that function looks like. So I would say take this message home. The soil microbiome does not equal biological activity. And here's some examples. So these are the kinds of common metrics that are reported if you have your soil microbiome sequenced. They'll predict nitrogen fixing, denitrification, all of the different nitrogen cycling aspects, phosphorus, et cetera, et cetera. And you'll note that they add this word called potential onto the end. And so that's kind of capturing this uncertainty. So the question is like, how well does the presence of an organism predict what let's say we know does nitrogen fixation? How well does that predict actual measured nitrogen fixation? And so this is an example of a study that is looking at the presence of key genes involved in each of these processes and if it's present, how correlated it is with the actual activity. So when you have an R of 1, it means if you have 5 of these genes, you have 5 units of activity. If you have 10 of these genes, you have 10 units of activity. They're perfectly aligned. And you can see that the highest we get is around zero point 0.7, which isn't terrible. I think as a researcher, you're always kind of happy that there is a signal there. There is some predictive value, but I would certainly not suggest that this is the most useful information in terms of what the microbiome is doing in your soil. So again, you could basically assess that this potential is a fairly weak signal to follow. And I have a funny example of this in a second too. So let's just revisit this. So the soil microbiome is not biological activity. There's only a weak correlation there. But as a soil microbiologist, I want you to take some heart in that we can make sense of what's happening in your soil using this kind of data. And that's because there are patterns in the microbiome that can be indicative of certain conditions. So let me walk you through what that looks like a little bit. So let's imagine that we have 12 different microbial species. Each of these different species, like each one of us, let's say, has a preference for a temperature. Some like it hot, some like it cold. So if you find more of one species that likes it hot, you might be able to suggest that this soil experiences more warm days on average. And surely enough, that is what we can find. And so you can do the same thing with different environmental parameters. Some of these same 12 species might have a preference for acidic soil versus alkaline soil. And you can start to sort of see that not all of those preferences align identically. So you can have an organism that loves cold temperatures and acidic soil, and an organism that loves cold temperatures and basic soil. So you're able to sort of pick out information, more and more information based on based on the patterns of where you find these different microbes. And the same can go for soil organic matter or any environmental parameter. Right. And so what does that look like in practice? So one of the most exciting bits of research I've generated in the last 3 to 4 years has to do with using the soil microbiome data, who's present in the soil, to predict different aspects of that soil. And one of the simplest ones was actually trying to use the microbiome data to predict the Cornell soil health test. So you can see on the bottom of these plots, you see low, medium, high, and very high. And that's what we're calling this health category. And that's what the Cornell Soil Health Test sort of summarizes as a general appraisal of your soil. And what you can pick out here is that each of these boxes refers to some oddly named bacteria in your soil. But the most important part is that you see a trend, right? In the lower soil health categories, you see that at a low abundance here, and this organism is actually much higher abundant in the healthier soils according to that soil health test. And you see the opposite trend here for these two. So I thought that was really fascinating. So I was able actually to predict whether a soil would be high or low soil health rated based on who was present in that soil. So that to me was a kind of an eye-opening experience. It's not that the microbiome doesn't have predictive value. It's just not as strong as we like it. And that's something that can change as we learn more and more too. The good news is that these different bacteria, I could actually follow up and ask, why would they be associated with a lower soil health? And it turns out that this group here are associated with nitrogen fertilization experiments in long-term fields. So when you add a lot of nitrogen to these soils, this specific group actually uses the nitrogen as an energy source. And so you see that typically it looks like that on average, farms that have lower soil health are probably adding more nitrogen. And that might make sense, right? That nitrogen might be lost more rapidly because they don't have a way of retaining it in field, et cetera, et cetera. Certainly, these microbes are happy that these farmers are adding more nitrogen. And the same goes for this other bacteria. When I went and read about it in the literature, it says that it's actually characteristic of dry, organic matter depleted soil habitats. Actually, many cases, desert and arid environments. And so you can see that some of these same farms that are applying a lot of nitrogen also have a depletion in organic matter, which we can predict based on this specific organism. So I hope I convinced you that there's some value to looking at the soil microbiome. I'm not saying it's all bad news, but my advice to you when you're considering whether it's worth your time, money, and energy to sort of go down this path is that is the company assuming that that genomic data or that microbiome data is equal to biological activity? Ask them that question. Like, what are your assumptions? And here's just a funny example of why that fails. So I did a test on my own DNA, and I found that I actually have a high performance athlete gene. So a gene that sort of makes your muscles twitch faster. That's common in Olympians, and I was quite pleased with this result. But then I went down to the local basketball court and I tried to dunk a basketball. And it turns out my genomic data was not predictive of my ability to put that ball through the hoop. And that's just a nice example. So if the company is making this claim, do ask to see the evidence. They should be able to show you some correlations like I showed you and give you a sense of how confident they are that they're predicting the actual soil properties. Also, consult with an independent researcher, right? Sending me or some of my colleagues at your land grant institutes an email, be like, hey, what do you think of this service? We can give you we all have our opinions on these different things. And I think we'll give you a pretty reasonable take on whether it's worth your time and money. So a lot of these are going for about $100+. But the key thing is, I'm actually pretty, as you can tell, I sequenced my own, genome, and I'm curious about this stuff. I think it does matter. It does have some value. But the key thing for you to retain that value is to ask for the raw data along with the report that you're given. Usually these companies will just provide you a PDF or some sort of paper report saying, here's what your soil looks like. I would encourage you to demand that they provide you the raw data along with that report because this microbiome data actually is good in the future. You can share it, you can reanalyze it. There's also all sorts of value. And the reason I say this is because the companies, their main business model is really to help build these large data repositories. The more farmers that use their service, the more their data grows and the better and more predictive it is. And that can be a net benefit to all of us. But the question is, are you just paying them to build this database? So get that raw data, and then if you want, I mean, there are people who are working on this stuff. Share it with an agronomist, share it with a researcher, maybe even shop it to another company that does this. They might be willing to pay you to get access to that data. In other words, that raw data is valuable. That's where the main value comes. Okay, so in summary, my take on the soil microbiome is that it's not equal to biological activity. We need to keep that at the forefront of your mind. It's an inference about that activity. But there is predictive value there. I am convinced. I've done that research. I can see that some organisms are really associated with different aspects of your soil. And if you find them there, you can say with some confidence that that's happening there. And then lastly, if you're curious, just make sure when you get that test done that you obtain your raw data. And that brings me to the end of my talk. I'd just like to thank all of the folks here in Indiana who support my research. Thank you for your ears. And I will now pass this off to Elena about these final slides. Thank
Perfect.
you.
Yeah, thank you so much. We have a lot of questions, so we'll get started here. If you are attending today to get a CCA CEU, go ahead and email me, Elena Whitaker at alenaw@iastate.edu by 5 o'clock today with your name, the name you entered to watch the webinar if it's different than like your Zoom username, and your CCA number. And we'll get those submitted for you to the CCA board to get some credits. You want to flip to the next slide, please? If this is your first time tuning in to watch an Iowa Learning Farms webinar for this year of 2025, you can go ahead and fill out our survey. It's totally voluntary. It's a demographic survey. You can find it by either scanning that QR code or going to iowalearningfarms.org/survey. It should take only like 2 minutes if you want to do that. And then I've got one more slide here really quick. Join us next week. We're going to have Andrew Hillman, who's a PhD candidate at Wisconsin-Madison, talking about investigating the disproportionality of nutrient losses in agriculture, the role of perceived vulnerability in conservation, and nutrient management. So that should be really exciting. With that, now we have a lot of questions. So let's get
Where to begin?
started here.
Yeah.
One of the first questions was, have you looked at the effect of biochar on the microbiome populations?
I personally haven't, but there's definitely some research out there. It doesn't, it's, biochar tends to be somewhat passive. It's a more passive form of carbon and an amendment than other types of carbon, at least from the microbial perspective. It's sort of a crystalline form of carbon, and it's hard for the microbes to get access to it, right? And that kind of speaks for itself because this stuff persists in soil for many, many, many hundreds of years. So it doesn't really, it's more inert than other forms. I don't know if that helps or what the curious question is, but yeah.
All right, this next question, how is the goop that you were talking about different from glomalin? Glomalin.
Uh, it's different because glominin is mostly produced by the fungi, of course, and that's a more protein-rich form of goop. I mean, honestly, there's a lot of different types of goop in soil, and microbes love to stick to things. That's one thing that they use to stick. It's another thing to help to protect. It's essentially their sheath. So it's harder for different chemicals or antibiotics or the bad things to get access to that cell if it's coated in this goo. It's a protectant. It also helps retain moisture. So most microbes in your soil are going to be producing some kind of goo. The goo that I showed that is produced mostly by the photosynthetic organisms on the surface tends to be more sugar based. So there'll be a bunch of sugars. It's sort of like your the starches in your pasta that stick to when you throw that pasta against the wall. It's more like the sugar. Yeah.
That kind of ties into our next question. They're asking, are the exudates only sugar, or are there also enzymes, proteins, vitamins, anything like that happening besides just sugar?
That's a great question. Yeah, yeah. The microbes are actually pretty good at sharing, it turns out. A lot of them will release when they're doing well and they're healthy, they'll be releasing things that are excess from their cells. And that will include vitamins, that will include other key molecules that others are quick to sort of soak up. And so one of the reasons why soils are so complicated to study from a biological perspective is that many of them depend on each other to grow. So we can't actually isolate them from a soil and keep them happy without knowing who their partners are that they share with. And so it's this very interesting and challenging question. For sure, these cells are pretty much leaky like the roots that feed them. So yeah, good
Cool.
question.
Our next question, they're wondering, does the improvement of the soil health triangle show grain yield increases immediately, or are they long-term benefits? And they say clearly like the
Yeah.
long-term benefits to soil health are clear, but they're wondering then about the
Yeah.
grain yield.
Yeah, I mean, when I think about the yield relationship with soil health, it's usually what's driving that is soil carbon. And there's some really good evidence that, if your soils are below 2% and you build that soil health and build that carbon, you will see a bump for the grain yields. But past that 2%, the relationship gets shakier. And again, there's different contexts. Context always matters. But if you're trying to think about the soil health yield relationship, it's mostly driven by that organic matter, and that's to do with its ability to retain moisture as well as provide nutrients, right? So it's giving those basics to the plant. But I will also urge, I mean, this whole soil health movement is not just about yield, of course. I'm sure you hear that as well. It's about that long-term fertility and activity of the soil itself, right? So people often are trying to are framing soil health in terms of resilience, right? Maybe you're not getting better yield one year, but because you have better infiltration, because you're feeding this sort of ecosystem that's reshaping that, engineering that soil, you might have better infiltration, better retention when there's a drought that year. Those kinds of relationships are harder, but it's also kind of where I always love to. Ecology to me is like economics, and proving a direct causation between some sort of social change or some sort of new technology and like an economic boost is tricky, right? And that's the same kind of challenge. So I think we'll get better at it. But I think the place to look is really these natural experiments where somebody's practicing soil health. There's some sort of stressor, whether it's a pest or an environmental stressor, and the soil health shows some benefits in that case. So that would be my framing of that question.
Perfect. Another question we have is do. They're wondering then about where did that question go? About basically like the timing. Is there a good time of during the growing season or any time of the year that they should be doing these tests if they want them done?
Oh, that's a good
And
question.
are the results going to change if they do it in
Yeah.
the spring versus the fall versus, summer, winter, anything like that?
Yes, they will definitely change. And that's because the availability of these soil resources changes, right? The most carbon-rich environment is at the start of the season, right? When all that surface litter is being decomposed and it's very rainy and it's washing it into the soil. You see a decrease in that respiration, that activity over the course of the season typically. And it's always correlated with rain. So when you have rain, you have a lot of biological activity happening. Um, how to handle that from a soil health testing perspective is to do it at a very similar time. Basically, these soil health tests, they're not always comparable across farms. They can be, but you basically what you're doing here is sort of benchmarking against yourself. From year one, if I do this the same time after maybe I follow my corn crop or I follow my bean, and I do that the same every 2 years, the exact rough conditions, right? So take note if you're doing it, what does the soil look like? Is it wet? Those kinds of things will matter. And that's the challenge with biology always is that it's like dynamic. The timing matters. But that's not to discourage you, but yes, yeah. Hopefully, as we study more and more of these soils, we sort of like pencil in what that looks like, and your data will be more comparable regardless of what time you take it. But we don't have those models yet.
Perfect. This next one, um, which soil health test do you think is the most accurate? Like the Cornell,
Hmm.
the Haney, any others that you could think of? Because they're saying there's a lot of them.
Sure.
There are a lot of soil health tests now,
Yep.
and they're all testing different parameters.
Yes. So the good news is that the Soil Health Institute did a very massive survey of the 36 most commonly reported soil health metrics. So I encourage you all to take a look at the results from that. They ended up saying that you need really three main ones that can predict a lot of what the other ones are showing you. Those were 24-hour respiration. So you put the soil in a bottle, you measure how much activity there is within 24 hours, similar to the Haney test, but not exactly the same. A little bit less work. The Haney test is good. The Haney test kind of captures both the biological activity as well as those resources that I was talking about. But the Haney test is not recommended by the SHI per se. They recommend respiration 24 hours, aggregate stability, and total soil organic matter. Those are the three that they think predicted most of the many tests that you can gain. And I really I trust their work and I respect their effort, right? Because they were trying to say if this is ever going to catch on, we need it to be a little bit more understandable, a little bit more relatable. So we can't have people do 36 tests on their soil every 2 years. So
Yeah, that'd be a lot.
yeah.
This next one, in the nitrogen enhanced or the fed soils, once nitrogen is removed, what happens? Do the families of the bacteria die off, and are they replaced then with other species that can handle a low
Yeah.
nitrogen environment?
That is what happens. You'd see some succession. Yeah, absolutely. You'd probably see a period of, hibernation. Microbes can withstand many period, like length of time until the conditions get better. But if they don't get better, you'll see some of those organisms dying off, just like we would kind of at the landscape scale that we're familiar with. You'd see some sort of change. Those organisms that are more drought tolerant will say, like you find in Indiana dunes or these arid environments will start to creep up, and the ones that need moisture will start to die back. Yeah.
Um, how resilient is the root microbiome towards disturbances and competition from bacteria that get introduced into the soil through
Good
manure
question.
fertilization or irrigation with, it says contaminated water?
Yeah. Oh, that's a fantastic question. I mean, all of these questions are really good. I'm really enjoying this. I think that's an open question. We don't know yet. There's been some really nice work on the introduction of fungi. So a lot of these arbuscular mycorrhizal fungi inoculants, they're not necessarily native to certain regions of the of our continent here, and you do see that they persist in some cases. Whether that's a bad thing or not is again to be determined. But we can see that the invasion ecology, right? This is the question about kind of relating to invasive species. Are we introducing invasive species that outcompete the natives? Is that a risk to our production systems, or is it maybe desirable because we manage these systems in a very contained, controlled way? Maybe having them in this specific place is a good thing because they match well with our crops. But I actually don't have a definitive answer for you. I think it's certainly one of the fundamental questions I'm asking now in the summer. I'm playing around with some bioinoculants. I've isolated them from 8 different fields in Indiana. We're taking those 8 bacteria and we're putting them all in those same 8 fields, and we're going to see, do the ones from that field outperform those that we introduced, and so forth. So we're kind of looking at those ecological dynamics of what we call invasion.
Okay, so one person is saying that they've seen increases in soil microbe diversity by changing their farming practices,
Sure.
but they're saying that farming is by nature extractive, and they're seeing a loss of various minerals in the soil.
Mm-hmm.
So, are there soil health practices that can be used to increase soil minerals?
Mm. I mean, certainly the biology in your field and your soil is your ally in that fight. But again, they're not always working as fast as we are, right? We have these very impressive plants that are very extractive and they do well. And there's no reason why we shouldn't consider augmenting minerals themselves, right? Like I in nature, you have very productive systems that are somewhat more imbalanced, but that's because we're not harvesting grain and we're not eating it. We're extracting those elements and putting them through human bodies, and those bodies are spread throughout the land, right? So there's a real tricky, wicked problem there. Um, I guess the question is it accelerating it at a meaningful rate? Are the microbes or what's happening in your soil improving that release in a way that has, a tangible benefit? And for sure it does, right? Like there's a lot of research coming out now, particularly with phosphorus, that we've been over applying phosphorus and that we're not really seeing many benefits from adding more because the microbes are cleaving it off, solubilizing it, returning it back into solution. So I would think that in that case, it's about timing. I'm certainly not I've never farmed at scale before, so take my advice with a grain of salt. I'm sure you are all much more familiar. But of course, like that early stage of development for that plant, that's when it's most delicate and weak, and providing it with a boost of nutrients at that pop-up stage makes sense to me. But as soon as that plant gets large enough to start leaking all that carbon into the soil and start feeding those microbes, you'd be amazed at how much nitrogen and phosphorus comes from the soil, even if you apply it, right? There's some really good work by Dr. Stuart Grandy over in Massachusetts, University, yeah, UMass. And he shows that, try as you might to add more nitrogen to the field in a synthetic or a soluble form, the plants still pick up most of it from the microbes. And that's because there's a timing problem, right? You're adding it at a certain time, it disappears or it gets soaked up by the soils, the biology or the minerals, and then it's not really accessible unless the microbes are actually cycling it for the plant. So yeah, I don't know if that answers your question, but yeah.
Here's a quick question. Someone was just wondering if you could provide the source for the rhizosphere video that you'd shown.
Yeah, I can. Yeah, reach out to me for sure. Yeah.
Okay,
It's a new Nature paper, yeah.
perfect. Um, Are there studies on how applied fungicides, pesticides, or seed treatments affect the soil biology?
Hmm. Yeah, that's definitely one of the common types of studies that you see. A lot of the data that comes to my mind has to do with the fungi because a lot of fungi are pests and we apply a lot of fungicides. And so the question is, does that have a negative impact on the fungi that are supporting growth through the roots? The good news is that a lot of the applications do not impact the root fungi unless they're systemic. So it depends on the compound. Does it get into the plant? Does it move through the vascular tissue? And is it translocated inside of the plant? If so, we do see that that has a negative impact on the beneficial fungi that live on the roots. But in general, a lot of these applications that are not systemic will not have a pronounced impact. We've got a lot of meta-analysis there as well, and so we know that if you do that study over and over again, unless it's a systemic fungicide, it's not very dangerous. Yeah.
Cool. Do, do, do. Someone is saying that they've tried the hanging test and also like the soil your undies test, where you're wearing cotton underwear and all that type of stuff on two different soils under two different management systems. And the results were counterintuitive. They
Mm.
had a higher SHC showed lower biological activity. They're just
Hmm.
wondering, do you have any insight on that? Any explanation or anything?
Yeah, I mean, maybe an example of how different microbes do different functions in the soil, right? So cellulose, those undies, that's not an easy molecule to digest, and that's for a reason. Plants essentially produce it as a protection. It's the part of the thick cell wall that prevents microbes from getting access to it. So it suggests, if you were to do that same test, let's say in where you put it on the undies on top of a log in the woods. I mean, I guess the log's pure organic matter, but it doesn't necessarily there's not a lot of readily accessible nutrients on that wood, right? The wood is also kind of a starved system. You'd see that those onions disappear, and that's because there's special fungi adapted on that wood to decompose that cellulose. So it might suggest that there's a mismatch between who's in that soil, even if there's a lot of organic matter, and their ability to digest. That's weird and that's rare, I would say. I think your case is a surprise, which is kind of interesting. But yeah, that might be why.
Okay, perfect. Um, I think I got most of the questions, at least that were in the chat, as far as I know. If you have further follow-up, again, yeah, feel free. Oh, one just came in. They're saying back to the fungicides. Do seed applied or soil applied fungicides have a negative effect?
Not from the data that I've seen, no. And again, just do a quick Google search, or maybe I don't know if I would trust the AI for these types of detailed questions, but it's the specific chemical that matters most, and ask whether it's systemic. You have to think that a lot of these pesticides, herbicides, they're actually kind of rich nutrient sources for a lot of microbes in soil. So a lot of glyphosate is one good example. Microbes chew that up because it's a source of nitrogen in the field and in the soil. So in many cases, they don't have a long enough like shelf life after you've applied them to the soil for them to persist to at a concentration that impacts the fungi. So yeah,
Okay.
the review for the various tests is from the Soil Health Institute. Um,
Yes.
it is published in 2019, I want to say 2020. No, I'm wrong. 2022. Um, but yeah, go to their website. That's one of their major products since they were initiated. Yeah.
Perfect. All right. You got to that question right as it came in, so that's great. Thank you. Raleigh for presenting today. Thank you everyone for attending. Again, join us next week for Andrew Hillman. He's going to be talking about disproportionality from nutrient losses in agriculture. So it sounds like a really great one. Again, thank you for all your questions. We had a ton of questions this week, so I
Great
really
questions.
appreciate it.
Yeah, thank you all.
Um, if you have any last words for anything, Dr. Willem, we'll take them now.
Just enjoy. I mean, biology is like I said. I mean, I started that presentation with all these interesting insights about how soils are special. I did this. Here's my own naivete. I went out thinking agricultural soils must be the best studied soils. So I went out into Indiana and visited these 14 farms. And I thought, we should know what's happening here. And my goodness, was I wrong. So much to discover in your farm fields. Like it's kind of exciting. Yeah, stay tuned. I would say that, you know, we should learn more over time, and we're starting to recognize the importance of soil and soil biology through the soil health movement. So keep trying new things, keep practicing good soil health. We'll learn a lot that way too.
Perfect. All right. Thank you.